I'll give a brief presentation of about 15 minutes. I hope not to extend myself too much. So, please confirm if you can see my PowerPoint presentation.
Yes.
Okay.
Thanks.
Okay, so I understand that these are chemistry students. So I'll try to speak. We are mostly from biology, except Professor Dr. Sonia Rodrigues. I'll speak a little bit about the potential of bacterial volatiles for bioremediation, and I will present a few of our works. So I'll talk mainly about bacteria and soil bacteria. So bioremediation in general is the use of living organisms to remove pollutants from or contaminants from soil and water. And we can also say air, because there are some research on that topic also. And usually most of the works are with bacteria and fungi, although of course we can use other organisms such as plants and animals. But traditionally, bacteria and fungi are the most used ones for several reasons. Namely, they are simple organisms. They can usually tolerate very harsh conditions. They can live with very limited conditions, for instance, carbon sources and so on. Bioremediation is a technology and an area of research which is very, very general, and it could be divided and is divided generally in several topics, depending on the organisms that are used or in systems that we are studying. But for instance, some of the main terms used in this area are phytoremediation, when you use plants. You can use plants to extract the contaminants or to degrade the contaminants. Sometimes you can try to do phytostabilization, in which you will try to avoid that the contaminants leached elsewhere. Or you can also use, for instance, rhizofiltration, in which you'll use plant roots to filter the contaminants. You can also use fungi. So in that case, you use the term mycoremediation. The term bioaugmentation is also used, and it means that we are adding the organisms, in this case bacteria usually, to the system. There's also this important term that is biostimulation, in which you will use the microorganisms that are already in the system, for instance, bacteria from the soil, to do the remediation work, but you will improve their growth by giving them nutrients, for instance. So this is an area of research that I think is very big and very, very diverse. It has lots of applications. And you can, as I spoke earlier, you can work with bioremediation in three main systems, either soil, the water, and even the air system. And the applications range from treatment of wastewater, from the mitigation of the effects of mines, and even recovery of the ecological status in mines. Sometimes it can be also used in industrial spills, for instance, in accidents, or petroleum spills with ships, for instance. Bacteria are mostly used for the petroleum spills because they can use hydrocarbons as carbon sources. But there is also some interesting research nowadays dealing with the remediation of agricultural soils, because they are many times they are under pressure of the use of fertilizers, for instance, intensive agriculture, And those fertilizers can contain contaminants, such as cadmium, for instance. And in this scope, you can usually remediate hydrocarbons, metals, fertilizers, pesticides, and volatile organic compounds. I would highlight the volatile organic compounds because I will speak mostly about them, but not in a perspective of remediating them, toxic ones, but on the role that they may have, on a beneficial role they may have. But there are some works trying to use, for instance, plants or bacteria to remediate, for instance, volatile organic compounds, which contaminate the air. Another thing that I think is important to speak, just as a background, is that there are many factors which will influence the bioremediation process, either biodegradation or phytosterilization or so on. And there will be temperature, oxygen, nutrients, pH, of course, the availability of water, and the bioavailability of compounds, also salinity and even pressure because of the reactions. I think this schematic illustrates very well, but I think there is a key thing missing on bioremediation nowadays, and that is the role of volatile organic compounds produced by microorganisms, so microbial volatile organic compounds, not in a perspective as contaminants, but as which is their role on the to be used as molecules that can aid us in this process. So what are microbial volatile organic compounds? Most volatile organic compounds of biological nature are more or less the same between organisms, although the microorganisms can produce a wide array of different molecules. Plants also can emit a wide array. And they can be, since you are from chemistry background, this is nothing new for you, but they can be from several families, chemical families. They can originate from fatty acids, and from fatty acids you can have the hydrocarbons, the alkanes and the alkenes, lipids, alcohols and aldehydes. Then we can also have amino acids that will come from we can have nitrogen compounds and sulfur compounds, which are originated from amino acids. Then we can also have some aromatic ones. Isoprene, for instance, is the most abundant volatile in nature. We can also have aromatic amino acids, just to give you an idea of the diversity of volatile organic compounds that can be released from microorganisms, okay? Many of them are bioactive. They can either promote plant growth, they can aid in the competition against other microorganisms, and so on. Now specifically talking about bacterial volatile organic compounds, there is some interesting research that has started more or less in the 2000s, 2003. Some of the works are regarding the bacterial metabolism, and I will speak mostly about this. But it's always trying to understand the role of these bacterial volatiles on towards the environment, and regarding the communication with organisms, be it competition or symbiosis, and they can have diverse applications. For instance, for biocontrol, and also as biomarkers. I will also speak a bit about that. But this is I think a very interesting topic of research, and which is more or less recent. So there is, I think that the bacterial volatiles have not found their way yet to technology of bioremediation. So I will talk a bit about our work on this topic, and I will briefly speak about a work that we did years ago in which we are trying to find how was the response of Rhizobium, which is a soil bacteria, to cadmium. And we tried to give a Volta perspective on that. We use metabolomics. I will not speak very much about metabolomics, as I know you'll have a speaker from Aveiro tomorrow who will speak about metabolomics. But we use metabolomics to provide insights on the response of the bacteria to cadmium, the soil bacteria, and to check if these metabolites could also be used as potential markers of stress induced by the metal. We used headspace solid phase microextraction, SPME, to extract the volatiles, and two-dimensional chromatography, coupled to time-of-flight mass spectrometry to check, to separate the metabolites and try to identify them. We picked Rhizobium for our study because they are bacteria that perform biological nitrification, which is a fundamental service to ecosystems. Also because this bacteria, being so important, already had its genome sequenced many years ago. And also because we know that their persistence is affected by exposure to non-essential metals. And finally, because plant-promoting bacteria, which is the case of Rhizobium, can promote plant growth and tolerance to biotic stress, and thus can, for instance, improve remediation strategies such as fictal extraction. So we basically exposed the bacteria to different concentrations of cadmium, environmentally relevant concentrations, and then we decided to study both the extracellular fraction and the intracellular fraction. And regarding some, I will just briefly go through the results. Regarding the endovolatilome, the volatile organic compounds present intracellularly, we found that in general, along with increasing cadmium concentrations, there is an increase in the emission of volatile organic compounds of the several families tested, namely the alkanes, the ketones, and the aldehydes, alcohols, and the terpenes, and the terpenoids, and the volatile sulfur compounds. We also observed that there was a general increase in the volatile sulfur compounds here. We highlighted some of them. Also in this monoterpene, the alpha-pinene. And here we observed a decrease in methyl thioacetate, and we found out that since this compound also originates from methanethiol, and this is the same with these volatile sulfur compounds. Since the bacteria were producing these ones, they were not producing methyl thioacetate. But just to see that the volatile organic compounds of the bacteria are changing. Their amount is changed because of the exposure to the metal. Regarding the exovolatilome, we will observe study the same, more or less the same families of compounds. And we decided to study the exovolatilome because it can be very useful as biomarkers of metals. To check, since it is non-invasive and we can, using solid phase microextraction, get an idea of how the metal is influencing the microorganism growth in their volatiles. Here we observe it again, in general, an increase of the production of volatile organic compounds. And we highlighted some of them. And in general, in this study, we observed a higher amounts of volatile organic compounds being produced as a response to cadmium, and we propose them there as potential biomarkers of cadmium-induced stress. We also believe that they are associated with several functions which might be of interest for the remediation of heavy metals. Namely, some of them we believe are involved in cell aggregation processes, the 2,3-butyrione, for instance, and 3-methyl-1-butanol, and several ketones. Cell aggregation is very important for bacteria to form biofilms and resist the harsh conditions imposed by environmental factors such as the oxidative stress imposed by metals, for instance. We also observed some other compounds being released, for instance, some antioxidant compounds, such as menthone, which originates from menthol. And intracellularly, we also tried to depict what was happening, but I will not go into detail. Based on that study, we decided to study, to further study the potential role of the conversion of alcohols into aldehydes. So observed here that in this study, that there was an increase in the aldehydes, and we observed this is a different work, but we observed that in exposure to cadmium with the same bacteria, aldehydes have the conversion of aldehydes and alcohols is they can go each way, but aldehydes are much more toxic. So we observe that reduction. This is actually, or this should be the inverse, aldehydes to alcohols, because alcohols are much less toxic than aldehydes, and we found that in this bacteria, their conversion would improve the oxidative status of the bacteria. Then we were also intrigued by the effect of the size of aldehydes on the response of the bacteria. And we found out that aldehydes with a higher carbon number induce a higher lipid peroxidation on the bacteria. So this is also something that must be considered. Even the same the same family of compounds can have different effects depending, for instance, in the carbon number. And finally, we also tried to understand which is the potential role of monoterpenes, which are usually antioxidant compounds, on the influence of the bacterial tolerance which is the influence of the monoterpenes on the tolerance of cadmium, of bacterial tolerance to cadmium. Again, using the same bacteria, we tried to study why did that increase in the production of volatile monoterpenes in that study that I spoke previously happened. And then we tried to, using an assay based in divided plates. I will, in a few moments, speak more about this, the importance of these divided plates assays. Try to check which was the influence of the biochemical endpoints. And we checked several endpoints, like in the previous work. I didn't speak specifically of each one. We can then, I know that there is a discussion session, so we can talk more about them, these biochemical markers. Usually they are either of damage, lipid peroxidation, or mechanisms to fight oxidative stress, namely glutathione S transferases, and so on. So we studied the alpha-penene, limonene, linalool, eucalyptol, and menthol. And we found out that there was, depending on the compound, we could have diverse effects. Some of them had positive effects, like eucalyptol, and others, like alpha-penene, could have some, at the same time, some interesting, some positive effects on the bacterial tolerance to metals. But also, at high concentrations, they could have, they could induce damage. So it really depends on the molecule. So there is not much research done in this topic, I think, on the role of the volatile compounds on bioremediation. That's why, especially the bacterial ones. So that's why I decided to speak about this topic. But I think there's a lot of potential. Usually when the few works that are happening with bacterial and volatiles are usually trying to check for growth promotion of the bacteria that they can promote the growth of plants. This in case it's in a control condition. But sometimes we can also try to check if they can improve the resistance of the plants to a disease, for instance, induced by a fungus in this case. But there are also some studies that are trying to check, for instance, the effects of the volatile organic compounds on the bacterial motility, and then in their swarming effects on the antibiotic resistance also. So I think it will be very interesting to see more studies on the role of the volatile organic compounds on the bacterial tolerance to metals, but also in their role, how does the volatile organic compounds or specific ones improve, for instance, the degradation or the bioremediation? Because we know they can have an effect on the bacteria and then their metabolisms based on the results that I showed you previously. So it will be interesting to see some in vitro studies using divided planes, for instance, with heavy metals, for instance, or pesticides or hydrocarbons, and to try to check which are the effects and if it is improved or not, since the research right now is very limited, only with growth promotion of plants and a handful of papers on the antibiotic resistance. And usually to study this, there are two ways to do it, either by capturing the volatiles using traps or using solid phase microextraction, which is, in my opinion, easier to do, like in the work that I showed you. I think that something that will be also interesting to see is the role of the synthetic biology. I don't know of any works that are working right now with this, with the bacteria, but I know, for instance, you can use the bacteria, for instance, and modify them with synthetic biology to work as sensors, for instance. So to make them change colors in a lake, for instance, if it is contaminated. So it will be very interesting to see how we can manipulate the bacterial genome, see an effect on the volatiles, and see if that will have a response or not on the bioremediation processes. But this is speculation. I don't know yet of any works that are, since we are right now only shedding light on the role of the volatiles, I think it's too much for now to ask for this, but I think it will be an interesting lead. Okay, so I believe that you probably have more questions than answers right now. I don't know if you want to make some questions or if we move to the next speaker, but probably a few questions if you want, and we'll move to another speaker. I don't know. So thank you.
Okay, thank you very much, Dr. Paulo. I think we will go with the next presenter, speaker.
Okay.
After
okay.
that, we will give a question and answer in the last session.
Okay, so thank you.
Who the next?
The next will be Professor. Okay, thank you. Will be Professor Italvina Figueira, and she will talk about diatoms as a bioremediation tool. Dr. Adi.
Okay,
please,
so
Professor.
I think that, on that part, it is afternoon. So good afternoon to everyone. Good morning to the side. I will present a work that we did in our laboratory. That is somehow we can use some of the results for a bioremediation approach. So the title that I gave to this presentation is Diatom Frostwings as a metal bioremediation tool. And I begin with a question that what are diatoms? For those ones that do not know what diatoms are, there are a major group of microalgae. They are found in oceans, also in streams and rivers, and also in soils. They account for 25% of all phytoplankton on Earth. They generate around 50% of oxygen produced on the planet. They also fix a high amount of CO2 from the Earth atmosphere, and they are characterized by having a silica-based cell wall that is named Frostwings. So, I was trying to. This is not going to the next. Okay, sorry. So, what are frostules and frostlings? So these diatoms have this silica cage where the cell is enclosed, and this silica is composed of several pieces, like you can see here. So you have like a lead, two leads, one on the top and one on the bottom. That the one on the top is termed epivalve, the one on the bottom hypovalve, and then there are some girdles that join these two leads together and make the frostule. And what are then frostulings? What happens is that there are some proteins that are enclosed, most of them, or some of them are enclosed in this frostule. That is, this frostule is made of a silica compound or a silica polymer. And then you see that there are several different types of proteins that are integrated in this silica cage. And so you have in blue silafines, then you have in green pleuralines. You also have some polyamines that you can see in this enlargement of this area. And all the frostule is surrounded by frostulings that are these red proteins that covers the outside, all the outside of the frostule, and that are maintained together by interactions with calcium. So you can see that these are these proteins. So these proteins, these different frostule proteins, have different domains. It is not important for us to see in detail each of these domains, but is just for you to see that the different proteins are quite different from each other. Okay, so then we also have metals on heavy metals, and they have a relatively high density. So I found that in the literature, numbers between 3.5 grams to 7 grams per cubic centimeter, they have metallic properties and they have atomic number higher than 20. They're also characteristics of heavy metals. Some of these heavy metals have biological functions. Some of them are macronutrients, like cobalt, like copper, like zinc and iron. Some of them are micronutrients, and as you can see, there are some heavy metals that are at some time macro and micronutrients, because that depends on the metabolism of the organism. So for some organisms, copper can be a macronutrient, and for others, it is a micronutrient. Also, as a the same happens. Copper. Then you have iron can also be either a macronutrient or a micronutrient, and then you have nickel and chromium as micronutrients. And then we have other heavy metals that don't have any physiological functions known by now, like cadmium, like lead, silver, or mercury. So the sources of heavy metals are quite different. So they can come from mining activity, smelting, metallurgical industries, corrosion, waste disposal, fossil fuel combustion, agriculture and forestry. These are some of the main sources of heavy metals. And they are known to have toxicological properties. They are very persistent. They persist in the environment, in the ecosystems for a long time. They can bioaccumulate and can biomagnify in the food chain. They can produce chronic and sublethal effects at low concentrations. They can induce acute toxicity in plants, animals, and microorganisms, and algae. Some of them can have synergistic effects, and they can have teratogenic and carcinogenic properties. In diatoms, heavy metals were shown to have to produce changes in the biodiversity of the communities, relative abundance alterations, also physiological and biochemical alterations due to oxidative stress, and also they produce deformations in the valves, in the fossils of diatoms. These deformations can be produced by different stresses. One of the stress that induce these alterations are metals. And in this figure here at the right side of the slide, you can compare two diatoms. The one on the right, the one on the left are have normal shapes, and the ones on the right are have normal shapes. So they are teratological forms. So how were the objectives of our work? Were to identify quantitative and qualitative alterations induced by cadmium into frostful proteins, analyze cadmium distribution throughout protein fractions, understand how these alterations result in a new cadmium tolerance mechanism, and to see if there is a possible application of this mechanism in bioremediation. So the methods used were first, we grew diatoms in two conditions, a control one and another one that we added cadmium at a concentration of 0.2 milligrams of cadmium per liter during 5 days. Then cells were harvested by centrifugation, washed, leased, and then the frustal fraction was isolated and purified. In this purified fraction, we made a sequential extraction, first with EDTA with agitation for 16 hours, then with sodium chloride and urea, and we obtained different fractions of frostwings. These different fractions were precipitated with TCA, with sodium deoxycholate, and with acetone. And then protein concentration, proteins were concentrated using desalting columns. So then with these concentrated extracts, we made an STS page that allowed us to separate different proteins in gels, in polyacrylamide gels with 4 to 12% of polyacrylamide. Then the bands were colored with comazibulum. And the results were the total frostful protein per extract. So the extract or the fraction that was extracted with EDTA, you can see that it was increased, a very high increase in the protein content. All the fractions increased in the presence when diatoms grew in the presence of cadmium, but some like EDTA and urea had a higher increases than those ones extracted with sodium chloride. Then when comparing control and cadmium exposed diatoms, the relative protein content was not changed, as you can see, for any of the extracts. The results from the protein separation by STS page you can see that 29 bands were identified, different proteins were associated to each fraction, and the number of bands were similar among the different extracts. Then we have the relative expression of bands, and what we saw is that 75% of the polypeptides showed an increase or an increase or a high increase in expression. And the expression of polypeptides in the EDTA and urea fractions were the most increased, as you can see. So then we also quantified the cadmium associated with each cell fraction. So the 97% of cadmium was allocated outside of the cell. Only 3% of the cadmium was inside cell, was intercellular. Most of cadmium was associated with frostwings, as you can see. This is the red, like 71%. And the silica enclosed cadmium was very low, was less than 1%. So most of the cadmium allocated outside was associated or to cell polysaccharides or to frostwings. And most of them were associated to frostwings. So as a conclusion, we can conclude like cadmium increases cell wall protein content. We also can conclude that frostwings increased in the presence of cadmium, and this increase was about 6 times higher. Cadmium was mostly associated extracellularly, so 90% was associated extracellularly, and 85% of cadmium was bound to frostwings. So continuing with conclusions, the results of this work point out to the identification of a novel tolerance mechanism towards cadmium, in which the frostwings can play the role of the major cadmium chelator, and so they have a very big impact in the tolerance of diatoms in environments with a high content of cadmium. Cadmium stimulated additional frostwing production, which bind metal reducing cadmium availability for uptake. So this is like a mechanism that increasing the frostwing amount of diatoms in the presence of cadmium allowed diatoms to tolerate exposure to cadmium by increasing this amount of frostwings, increasing the binding of metal extracellularly to these frostwings, and making less free cadmium available for internal or internal uptake. So these results can be used as the new bioremediation tool, because they provide knowledge for the development of a field strategies using diatom biofilms to collate metallic ions. So this is our conclusions. I concluded my presentation. These results and other more, so I just presented some of the results. If you have interested or you can see these results in this article that is already published for some years ago. So thank you. I don't know if you have some questions. If you have, I'm glad to answer them.
Okay, thank you very much, Professor Etelvinda. Maybe we will discuss in the
at the end. Okay.
The end of the session. Yeah. Okay. And then maybe the next speaker. Who next?
Hello. I think I will be presenting next.
Okay.
I was
okay. Please.
For to sharing the screen, so I can start sharing mine. This will be quick. I think you can see my screen now.
Yeah, perfectly.
Yes. Yeah.
Thank you. So again, hello everybody. It's great. It's a great opportunity to be here today. And what I'm bringing you for this presentation is a more system view of the soil recovery processes and the soil plant systems. Also with the focus on metals and the metal availability in contaminated soils. But I will just start the very short and general overview of the system that I want to describe today. So as probably you well know, soil performs very important and relevant services and functions, environmental functions. And basically it acts as a very important filter and buffering system for all the chemical compounds and chemical elements that are deposited in soils. So whatever pollutants or nutrients that are input and deposited into the soil to complex biogeochemical processes, which I will not analyze in detail today, but I want you to keep this in mind, this idea that soil is a very important buffer and filtration system, as well as the place for many physical and biogeochemical transformations to occur. So once these contaminants or nutrients are deposited in soils, this will go through these processes, and very importantly, there will be a relevant share of the pool of elements which are deposited in the soil between the solid matrix and the solution matrix. The solution component of the soil solution, so basically is the water in the soil system that leaches and percolates in the soil pores and among the soil solid particles. This will play a very relevant role in making the chemical elements or chemical compounds that are deposited in the soils eventually available for uptake from plants or from organisms. So all this system can be disrupted by human activities, as you know. There are several threats for the functionality of the soils and and that may impair this capacity of soils to perform these functions, including soil pollution or contamination, which will be the key threat that I will be focusing today. So once you have contaminated soils from all these different sources, and particularly from contamination from mining operations, as I will bring you an example of applied research that we developed in our group, which relates specifically to the contamination and pollution of soils in mining areas and how to recover those degraded soils so they can effectively perform their functions and allow plants and vegetation to grow. But before going to this aspect, before going into the details of these recovery processes, I also would like you to understand that when soil is contaminated, particularly by a metal, when you have metallic contaminants in the soil, the bioavailability of this metal in soil, for example, I have here the example of lead, will be dependent on the mineral form of this metal in the soil, but it will also be affected by a series of transformations in the soil that will determine whether the element being present in the soil will be available for uptake by plants or organisms. So, and one of the key aspects that will control this bioavailability in the soil is not only the chemical availability in the soil, which will be affected by soil properties. And this is another key message from this talk, is that the physical chemical properties in the soil, the pH of the soil, as well as the content and composition of organic matter in the soil, as well as the texture and all the soil properties that will determine its cation exchange capacity is crucial when you are dealing with soil contamination, especially with metals. And it is this cation exchange capacity, because for metals, the metals that I will be focusing today, which are mostly cadmium, copper, zinc, and also lead, this will be present mostly in a cationic form in soil. So the way we have to control the bioavailability of these metals in the soils once they are present, for example, in mining areas, is not only to remove the actual metal by treating the soil, but also another possibility is to add amendments to the soil, which will control and which will contribute to immobilize the metal in the solid matrix. Because only when the metal will be then dissolved in the water solution, it will be available for organisms or plants. So what we want here, and what I will show you with our example of applied research, is that by immobilizing the elements in the solid matrix, with even without removing it from the solid matrix, we can already significantly improve the soil functionality and its capacity to support, for example, plant growth. So I will not go into many details on the processes that control this availability for plant uptake of metals in soils, but you may check the literature, and I'm happy also to send you additional information afterwards if needed. So as I said, we have an example that I would like to share with you today about an applied field test in which we use materials to recover mining soils and to control the plant availability of metals in soils. So what did we do? This relates to a live project which has been funded by the European Union, but it has also several partners. It's coordinated by Universidade de Aveiro, but there are also industrial and other academic partners from Portugal. And what we focus on this project is on the recovery of degraded soils in mining areas. Just to give you a very brief overview, we have even more than 175 degraded mining areas in Portugal, and especially those located in an area called the Iberian Pyrite Belt. The levels of metals and the acidification in the soil is very severe. So if you know, in mining areas, you have acid mine drainage, which will bring the pH of the surrounding soils to very low levels. So what we face in these specific mining areas is a situation of soils with very high levels of heavy metals, but also very low pH. The soil pH in these areas is quite often around 3. I don't know if you have an idea, but for ideal plant growth, for most plant species, the pH range should be between 5.5 and 6.5, or between 6 and 7. So this is what happens is that with such a low pH, you don't have the plant growth, because the metals will be mostly in the cationic and in the solution and extremely available for plant uptake. So we need to treat not only the metal contamination, but also the acidity of the soil. So what we did, in order to do this, we prepared amendments for the soils. And what type of amendments? These are biomass ash amendments, which is, what is this biomass ash? Well, it's ash that results from the combustion of residual forest biomass from one of our industrial partners. So this residual forest biomass was burned for producing energy. So we use the ashes to produce a soil amendment. And why did we use this? Because this is a very and highly alkaline material with pH around 10, 12. So you see from one side, we have this very acidic contaminated soil. On the other side, we have this residue, which is treated as a waste because it's the ash resulting from energy production from burning a residual forest biomass, which is highly alkaline. And it also contains nutrients from the biomass that was burnt. And because this is for residual forest biomass, the contamination levels in these materials are very low. So mostly you have nutrients that you can add to the soil, but not so much contaminants like, for example, you have in ashes from coal or other materials. So what we wanted with this project was to produce a soil improver and then to demonstrate through laboratory and field tests that this soil improver based on ashes would work. So we did several studies to characterize the soils in these areas. And then we prepared these materials. Being an ash, it cannot be applied directly to the soil, also because the particles are very small. We developed and optimized a pre-treatment and granulation process for these materials. I will not go in details, but I can give you some additional information afterwards. So we developed an actual soil amendment from these ashes that first we tested in the lab. And we ran the series of physical, chemical, and also biological characterization in the pot and column experiments. And again, you see here, and I would like to highlight again the role of analyzing the changes in physical, chemical properties of the soil once these materials are applied to then understand what will be the effect on the improvement of soil properties, on making the soils capable of supporting plant growth as well as organisms growth, because as it is, pretty much nothing grows. So as you see here, we had very interesting results, even when we did the tests in the lab, and I'll just give you some pictures here for which you see once the soils were amended with these materials, this could effectively support plant growth. So we did tests both on the soils and on the plants. Again, I will not go into much detail about this because we have a very short time, but I can give you additional information afterwards. So what we did after this was we went to the field. We went to an actual mining site. We prepared test plots, as you can see here. And as you see at this mine, these streams are very acidic acid mine drainage. So all these are tailings and wastes from the mining processes, which are severely affecting all these soils. And as you see here, no plants grow on this area, almost no plants grow on this area because the soil is affected by this acid mine drainage. So here you see how the preparation of the field for our tests. And then we actually prepared, we went from lab scale in the preparation of these amendments to field scale. So we actually prepared almost 20 tons of granulated ash. We also applied composite biological sludge to bring additional carbon inputs to the soils. And after a series of preparation of the grounds, we implemented 12 test plots in May 2018, and we applied the materials on these 12 test plots. So we had 3 soils, and we had 3 types of amendments based on this ash, biomass ash that we applied into the soils and that we now have been monitoring for 2.5 years. We also did almost 1 year after the amendment and the treatment of the soils, we sowed the soils with plant seeds because we wanted to, and only in our test plots, only half of the plots were sown with these seeds, plant seeds, because we wanted to check both the capacity of the vegetation to grow in a spontaneous way, and also we wanted to see that if we applied the seeds to the treated soils, whether these would grow or not. And as you see here, we had a very quick response. And only 1 month after the sowing process, we could clearly see that in non-amended soils, which are the ones you see here, so the soils, the mining soils with no ash amendment, you don't see plant growth at all. But in the plots that where we placed the seeds, in the out of the plot with the seeds, you see these more homogeneous growth of plants, because these are the sowed areas, but you could also clearly see that the vegetation could develop also in the non-sowed half of the treated plots. And you see here very clearly that 2 months after sowing, so again, these are pre-control plots in which we didn't added our soil amendments. But for the treated soils, you clearly see that especially only 2 months after sowing, you could already see very clearly the plant growth and the plant development. Again, for these soils here, which were the three control mining soils, pretty much nothing grew. So then what we are doing now is to understand and describe and characterize what were the effects in the soils and in the soil plant system that enable the growth of plants. We are using this information for optimizing future applications of materials in the soils. So just very quickly, I will just very quickly present you a few results. So you see here for three mining soils that we used, these low pH values are the three controls, and you see that the pH correction was quick and very effective. In fact, nowadays, 2.5 years after the application of amendments, and this was one application only, we still have the pH values, these values between 5.5 and 7.5 in some of the cases. The same occurred for organic matter, of course, for the organic carbon. Of course, in the plots that we amended with also composted sludge, the increase organic carbon notorious, as well as nutrients. So if you here on the left, you always have the controls, and then you see the increase also in the nutrient soils in the treated plots. Just one of the final key messages here, I was telling you about the importance in correcting and improving the soil properties to control the bioavailability of the metals in the soil. So even though we did not remove the metals from the soils, we developed a system which allowed to immobilize the metals, the contaminants in the solid matrix, as well as the nutrients and adding the nutrients in the soil. And you see here, for example, the copper, the example of the copper, which has very high total levels in these soils. And these are extracted contents with dilute acids, as you see here, just in blue. So you see that in general, the presence of copper is similar in all the plots. So for the three soils here, you see the blue bars, which represent the reactivity of the metal in the soils in relation to the total. So this will be the total pool that would be reactive and available to the plants. But then when you look into actual available metal in the pore water, which are here the color bars, you only see that in the non-amended plots, which are these three, A0, L0, and SD0, there is an effective presence of dissolved metal in the soil solution that will impair the growth of the plants. So for the treated 40 plants plots, we were able to remove this availability. And this was traduced not only in the growth of the plants, but in the improvement of the soil as a system. You see here again, just an example which shows the clear increase of colony-forming units. And this study is being performed also with Professor Albina and Dr. Paulo Carduso, which just presented before me. And we are now focusing on better explaining these processes to fully present the picture and characterize the picture of the improvement of the soil function and functionality. And the relevance of this is that you see here that this picture was taken around 1 year ago. What happened is that these mining areas have very dry and hot summers. So during the summer, the vegetation in these plots and in these areas tends to dry, and then it grows again in the autumn period. And as you see here in our plots, and this was confirmed again this year, is that we were able to reestablish the plant growth, and we were able to reestablish and regenerate the system. From the three amendments that we used, we realized that the use of the ash granules that we prepared with the compost was those with an impact, a more effective impact on soil properties that also allowed to support plant growth, as well the development of microorganisms and organisms in the soil. We also have additional tests, which I'm not bringing to you, about, for example, the growth of invertebrates and the reproduction of invertebrates on these soils. So basically, by correcting the soil physical chemical properties, by adding these amendments, we were able to recover the capacity of the soil to support life, meaning life from organisms and life from plants. And this is crucial, especially if you have this, and this will be my final message, if you have in mind that the population growth worldwide, the population is growing very fast, and that by 2050, we expect to have around 10 billion people in the world. And in order to produce food to feed this rapidly growing population, world population, we will need to produce, obviously, to have soils that are capable to support growth. Nowadays, with the current highly intensive agriculture practices, we are degrading our soils. And with all the industrial and human activities that degraded the soils that we have, we will need to be capable in the future to very quickly recover many areas to make, and many soils worldwide to make these capable of, again, supporting plant growth and where needed to produce food for this growing population. So thank you very much for your time. I'm happy to give you more details about these studies and to answer questions where relevant. So thank you.
Okay, thank you very much, Dr. Sonia. And for the next speaker, please.
Hi, everyone. I think that the last one is me. So let's put my test.
Okay, please.
So, okay. I think that everyone can see my presentation.
Yes, yes.
Okay, I'm Adelia Peach, as Paul presented me. I mainly work with polychaetes. And so I will speak about that group of organisms that are mainly marine and live usually inside the sediment. So I will explore the use of these species, of this group of species, in bioremediation of contaminated sediments. So polychaetes are the most abundant group in benthic communities, are important species in estuarine and coastal food webs, and usually affect the biogeochemical cycle of nutrients as well as contaminants availability. Also, some species are economically important because they are used as fish bait, or also they are used as feed in aquaculture systems of fish and crustacea. Human activities have increased the number of and concentrations of contaminants that enter in natural aquatic systems. Some of the most usual contaminants include metals, hydrocarbon compounds, and organic matter. These contaminants may accumulate in sediments and then will have negative impacts across multiple ecological levels, and also may interact with other environmental variables that can cause synergistic or antagonist impacts on aquatic systems, structure, and function. Thus, bioremediation of these contaminants is very important and is required to maintain the ecological balance, the biological diversity, and sustainable development of aquacultures. One way used in remediation is the use of marine polychaetes, usually bioturbators, that are the organisms that more interacts with the sediments and have more impacts in sediments characteristics. And so are usually polychaetes with this behavior are used in bioremediation of marine sediments. Bioturbator species. Why bioturbators are important candidates in the remediation of contaminated sediments? Because the species with this behavior usually reworks the particles, bioirrigates the sediments, and also other organisms will benefit of this behavior. The sediments are more oxygenized, and so more organisms are able to live in this type of sediments. Additionally, this behavior, this bioirrigation behavior will affect and mobilize the contaminants, organic contaminants, by increasing the transport and mix the superficial water with the sediments, improving the pore water well, at the end, the sediments is more homogenized and more aerated. Also, bioturbators will increase contaminant sequestration by the formation of complex biofilms derived from bioturbator organic waste. And the boring activities will increase benthic metabolism and nutrient dynamics by stimulating aerobic material communities that will participate also in contaminants degradation. I will start to present you some studies with sediments from aquacultures. Usually in fish and crustacean aquacultures are produced large amounts of uneaten feed and feces. And these organic materials consist mainly of organic compounds like carbon and nitrogen and phosphorus that usually are present in the diet such as proteins, lipids, carbohydrates, vitamins, minerals, or pigments. And the accumulation of these organic waste will lead to a logistical problem in the aquaculture's facilities, because will cause organic enrichment, will reduce dissolved oxygen concentrations, and will increase the levels of acid volatile sulfides in the sediments. And this will lead to negative impacts of other organisms and also in the water of the aquacultures, in the quality of the water. So there are several studies that already use the bioturbator polychaetes, and they will have an important role in the recovery of the sediments in aquacultures, because they will influence the nutrient dynamics, increasing organic matter breakdown, and that will lead to the release of ammonia and phosphorus to the water column. And also the sediment oxygen uptake will be enhanced with the presence of these organisms. Studies carried out with the two polychaete species, Aedes versicolor and Capitella, demonstrated that the mineralization of organic matter from sediments of fish aquacultures during 2 months, this percentage of degradation was 135 for Aedes and 87 for Capitella. As you can see, is a very huge contribution for the organic matter degradation in this kind of sediments. Other study carried out with Biochemical Markers. This study has the objective to understand, to try to understand the use of these species also in an aquaculture, in a local aquaculture. And so they exposed different densities of the organisms. They have several groups with different densities, and the authors observed that after 45 days of exposure, this polychaete, Biochemical Markers, was able to remove at the end, at the highest density, 200 organisms per square meter, they have the higher rate of organic matter removal. Also, at higher densities, the total nitrogen and carbon significantly decreased after 45 days of exposure. Here I have another study that intended to compare the behavior, the bioturbation of 3 annelids of 3 species. And here you can see the control, so the sediment also rich in organic matter that it was not exposed with the annelid. And here you can see that the sediment have several layers. So no, it didn't occur any mixture of the sediments. And here the sediment, here is on the first day, and here is on the last day, so not much difference between the after 45, 50 days in this case. Here in the exposed sediments with annelids, you can see here in C image some burrows, but in the last one, E figure, we saw that this polychaete, that it was Perinerisch nantia, have a higher irrigation. Here you can see more burrows, and also the sediment is more homogeneous. The studied species, the 3 species, had different sizes. These ones, Thalassodrilich and Capitella teleta, are smaller species. And Perinerisch is a larger one, and as you can see, he's more efficient to remobilize the sediment. Here, another is the same study. They also quantified the sulfites and put the redox potential, that it was low at the end of the exposure for Perinerisch, but also for Capitella. You can see here. These results indicates that ventilation and irrigation behaviors of these polychaetes was very important to lead to the introduction of oxygen-rich water into the sediment, conducting them to reduce the levels of sulfites and increasing the redox potential. So there are actually several culture systems, aquaculture systems, that have already integrated polychaetes in the sediments with the objective to remobilize and to degrade this organic waste that is characteristic of these systems. Additionally, these groups of species, these polychaetes, are important source of proteins and lipids, and so they again can be used at the end as feed for fish or crustacean that are used in the aquacultures, and so is a more at the end is a more efficient system. Now I will show you some use of some species of polychaetes in bioremediation of contaminants. In that case, the polyaromatic compounds, hydrocarbons. As you know, it's one of the most abundant contaminants in the aquatic environment. They are very toxic for organisms. And so the remediation of this contaminant, it's important, and it already occurs in the marine sediments. And several studies already reported use of polychaetes in the bioremediation of this kind of contaminants. Here I have again the use of the 3 species that I already spoke in organic matter enrichment. I have Thalassodrilich, Capitella teleta, Perinerisch nantia, that were exposed at sediments with high concentrations of polyaromatic hydrocarbons, and after they were in the sediments, in these contaminated sediments during 50 days. And at the end, the authors observed that the sediments had less 56, 55, and 68% compared to the initial levels of these compounds. Also, other studies reported the removal of 63% of fluorine in sediments where capital atleta was introduced, and the same study reported the use the sediments where Adi versicolor was used. 25% of pyrene was removed of sediments exposed. These three experiments or works that I spoke at with these species occurred in the laboratory, and it's well, the main studies with this kind of contaminants are occurred still in the laboratory. There are not so many studies in the field. So, however, these results confirms the ability of this type of species of polychaetes to be able to be used as bioremediators in contaminated sediments in natural environments. But how these polychaetes are able to bioremediate this kind of contaminants? Several authors reported the presence of enzymes of phase 1 and phase 2 in polychaetes. It was already observed in the genome, and how these enzymes are able to biotransformate the polyaromatic hydrocarbons. And also the polychaetes are, and this is maybe is the main factor that it was pointed out by several authors. Polychaetes, by bioturbating and bioirrigating the sediments, will improve the capacity of these sediments to be able to install, to enhance the microbial community that are able and more suitable for degrade this kind of contaminants. So they are these polychaetes can degrade the contaminants, but their behavior, it's very important to enhance, to improve the microbial community that is more suitable to degrade polyaromatic hydrocarbons. So to finish my presentation, organic matter and nutrients in the study of this kind of contaminants in the sediments, polychaetes were very well studied. The majority of the studies is with this kind of contaminants. And the families that were reported to be more efficient to remove organic matter and nutrients are the Erniclids, Capitulidae, and Nereidae. In the case of polyaromatic hydrocarbons, the majority of the studies included Nereidids and Capitulids. Capitulids has also the most efficient families. But the quality and the degree of these organisms' effects on sediment properties will depend on the organism's biomass. As I told you before, the density of the organisms matter. Higher densities is more efficient to to degrade organic matter, for example, and also life strategies of the organisms. For example, large organisms are usually large organisms and more and active bioturbators are more effective in mineralizing, degradate mainly organic matter and nutrients than smaller species. As future research directions, it is important to increase the number of field studies, as I told you before, mainly for polyaromatic hydrocarbons. The majority of the studies are carried in the laboratory, so it is important to apply this knowledge in the field in contaminated sediments. Also, it is important to consider other environmental variables. As we know, in the environment, we don't have usually only one contaminant. We have several contaminants and also other variables that could have synergistic or antagonist effects, and thus the knowledge of these variables interaction should also be considered. And at the end, so as you can see, bioturbation that leads to, it's possible to be used to lead remediation of contaminated sediments of marine habitats. But it is important to develop a more specific approach using adaptive management approach. Because why I'm saying that? Okay, I spoke here about several families that are usually more efficient or less efficient by removing contaminants. It is important to use local species, native species, to avoid introduction of other species that we don't know which impact will have in the new place. So it is important to study the local species and explore their use for this kind of remediation. So thank you for your attention, and if you have any questions, please.
Okay, thank you very much. We got the 4 interesting speaker here, and okay, I will give some overview. So Dr. Paulo give us the topic about the bacterial response in the presence of the cadmium, and Professor Etelvina give response also for cadmium to increase the protein and in the frustulin. And Professor Sonia also give us the different perspectives, not in the library scale, but in the pilot project with the real problem in the mining degraded soil by using ash to promote the bioremediation. And the last, Dr. Adelia give us the biodegradation of organic matter by using polycate. Okay, I show in the chat we got 2 questions. First for Professor Sonia, I think, from Taufik, asked about the best treatment for the mining problem in the Tarakan, and Professor Sonia had answered also in the chat, but she used fly ash for the treatment, and for the detail you can check the publication. Also, she give the link.
Yes,
And
if I
yeah.
may add, just very quickly, I
Okay.
may just enter just very quickly.
Yeah, please.
So, I included a link for the website of the project, as well as a few publications. In the project, you have the website, you have the list of all publications that you may check. So just very briefly for this material, these were produced from the thermochemical conversion of the residual forest biomass to energy. So basically, when you have the residual forest biomass, for example, from cleaning the forests to prevent forest fires, these are kind of materials that can be used to produce energy, and that are use energy. And in one of our partners, the industrial partner, which is the pulp and paper industry in Portugal, they use their residual biomass to produce energy. So then they produce these tons of ash, and specifically we use the fly ash that we developed this granulation process with water. So these granules, hard granules of ash, which have been stabilized for 6 months before application in the field. So basically you have large quantities of calcium oxide, which by the stabilization process convert to calcium carbonate, and as well as many other minerals that I will not go in detail, but that then will react in the soil once it applies in the soil. So the question about how what is the matrix that immobilizes is the actual solid matrix in the soil that retains and immobilizes the contaminants by the change in the soil properties, and also by the addition of the control of the acidic pH. So these elements will only be in the soil solution in the presence of acidic pH. And as for long, once we apply these materials, we observed results right after that. 2 weeks after the application, we already observed the results, and for 2.5 years now, and with one application only, the treatment is still effective. But you may check the publications for all the.
Okay, thank you very much. Okay, maybe anyone for any question? Before in the chat, we got the question for Dr. Adelia from Miss Diana. How to increase the population of polychaetes, mainly if we can, if we want to apply for the bigger pond, because in the case in her place in Tarakan City, the pond that is for agriculture is very wide, about 1 to 5 hectares. Okay, please, Dr. Adelia.
Okay. What I could say is before you should study the community, the benthic community. Not only polychaetes are used for bioremediation. Also other bivalves, for instance, or other mollusks are also used, or crustacea. So first of all, investigate study the benthic area. So for other species, and then you should investigate, you should study or research how these species reproduces, and reproduction is the best option. So, but you should see which species are more abundant, and the ones that could be bioturbators to be used for these as bioremediators. for example, here in Aveiro, one species that is very used is Aedes diversicolor, is a native species, and so is very used here. But it was already did a study characterizing the benthic fauna, and then they studied the reproduction, how to reproduce these species in the laboratory and to improve this the reproduction rate. And then they are using these species in aquacultures.
Okay, thank you very much. I have a question for Dr. Paulo. So you presented the bacteria can tolerance with the cadmium. So what happened with the cadmium? They only accumulate in the bacteria cell, or maybe they release in the environment after modify or something?
Well, actually, for the bacterial strain that we were talking about, it was that strain is a Rhizobium strain, and it's Rhizobium E28. This strain actually is capable of accumulating the cadmium and tolerating its presence intercellularly. Professor Tolvina can also shed some light on this, because we have been working with this bacterial strain for many years now, not only regarding the volatilomics, but also regarding the biochemical response. And when comparing the E28 and another strain, a bacterial strain, a sensitive one, we observed that it actually accumulated a lot of cadmium, but it was able to tolerate it, to fight it with the antioxidant system, the enzymatic one. But I believe if Professor Tolvina wants to join this question, she can develop further.
Okay. So also I think it's related with the diatom, and then because the same.
No, I'm not talking about diatoms. I'm talking about bacteria.
Oh, you had your microphone muted.
Yes,
Professor.
yes.
Okay.
It's okay now. What we already studied is that these bacteria, when they are the most tolerant bacteria to cadmium, were the ones that accumulated more cadmium intracellularly. And what we did is to then was to understand why this happened, and what we were happily find is that these bacteria that can accumulate, are more tolerant and can accumulate more cadmium, that are able to produce a higher amount of glutathione, that it is a tripeptide that is a very potent antioxidant, and this glutathione tripeptide makes complexes with cadmium and neutralize cadmium effect. So cadmium no longer is free in the cytosol, and because it is complexed with this glutathione, it does not interact anymore in the metabolism of the bacterial cell, and then it allows cells to tolerate higher quantities of cadmium outside, and they also accumulate inside, but they are neutralized, and so they allow bacteria to tolerate this cadmium. That's what we find out, and these results are also published. So if anyone has interest to read, it is available.
Okay, thank you very much for explanation. Okay, we got another question for Professor Tolvina. Is diatom frustulin can be used to collecting another heavy metal except the cadmium?
Yes. Yeah. So these frostwings are not specific for cadmium. They can also because this interaction is a charge interaction. So these frostwings have these cysteine. These cysteine groups that have cysteine-rich domains, that have these sulfidyl groups, TDCTO groups, and then they can interact with any metal that has a positive charge. So they can interact with any metal. Okay.
And also, if we want to improving the capacity to larger scale, what we do next for optimize and also
if we want to increase the production of frostlings, as you already saw, when the diatoms are exposed to the metal, they increase the amount of frostlings. There is no work to see if other conditions can improve more these frostlings. There's work to do over there, but I cannot answer that because no one did it. So I cannot tell you.
Okay.
Okay.
Yeah. Thank you very much. Is there any question? Okay, I have one related with the pH gradation. So, did you identify the metabolite from the degradation of pH?
No.
Yeah,
I did not, but there are some studies that yes. There are studies that reported the products of degradation. Yes, I did not, but if you find, I think that one of the authors that I cited, one of I think one of them quantified and identified the metabolite, but I'm not sure. But if you want, if you are interested, I can see after and send you the paper where I saw that. But yes, there are authors that already identified it.
Yeah, because the pH is the big molecule, so it should be degraded to simple.
Yes. I saw one I think that it was with a dist, I'm not sure, where fluorine was degraded in three subproducts. I don't know the name, but it was divided in three simpler metabolites.
Okay, thank you very much. Okay, is there any question? No. So I have a question also for Professor Sonia, that because in some case, S also include as waste also. So I just worried that maybe in our country, S will be treatment by in different more safe product, because as also byproduct by some mining also. So I'm worried that also why you thinking about the S also will be contaminant in the soil.
Yes. So yeah, of course, you have a very important point here, and that's why we perform this long-term study to evaluate also all the potential risks associated, which is a key aspect here. But be aware that one of the references that I included, it specifically relates with the properties of the ash and of the origin of the ash that may be used for this type of application. I highlighted in the presentation that this is residual forest biomass ashes, so not from coal combustion. So the type of material that is used, the type of fuel that is used, will control the elemental composition of the ash. For example, if you burn, let's say, wood that was removed, that was used for construction, or painted wood. So if you use contaminated materials to begin with, then yes, you have contaminants accumulation in the biomass. For example, reports about cadmium and lead contamination in ash from treated wood that was treated with specific materials. So it is very important that you also characterize the type of ash and the type of biomass that was burned in order to control this and the chemical composition of the ash. But as our results demonstrated, by using general, and also there are specific types of trees that may also contribute, for example, to increase not the levels of contaminants, but for example, the levels of chloride, which is also a key aspect for the composition of the material that you use. So this is another aspect that we are focusing on, which is in controlling the levels of chloride, as well as other elements in the ash that you use. And then our pre-treatment process is also aiming at improving the composition of the final granules that will be applied in the soil in order to prevent any risks from the potential addition of contaminants.
Okay, thank you very much.
So it is key if you characterize the material. You need to know your materials for applying them, but once you do this, and we are producing guidelines on how to characterize these materials, then you can safely, more safely use these materials in remediation applications.
Yeah, okay. Thank you very much. Is there any question? No? If is there no question again, so I will finish the discussion today. Thank you very much for speaker from the University of Aveiro, Portugal. So I send back to Dr. Yuli and Dr. Kiki. Thank you very much.
Okay, thank you. Thank you for all the speakers. We have a very nice talk this afternoon or in the morning at Portugal. So for all the participants, don't worry. I have recorded this lecture, and I will upload on the chemistry department YouTube channel. So maybe you can subscribe our channel. So if I upload the new video, you will get the notification. Okay, I think it's enough for Dr. Kiki. You have anything to deliver?
Okay. Thank you very much for Dr. Paulo and team. It's such a very interesting talk. Hopefully, we can have more collaboration between IPS and some other university in Indonesia with Universidade de Aveiro. Hopefully, on next semester, we have more online class.
Okay,
so, yeah.
Okay, that's Dr. Kiki said. I hope in the next semester we can continue this lecture and maybe the others collaboration, the other activity. Okay. Thank you again for your time and for all the lectures. Okay. So, and for all the participants, we also say thank you for your attendance. Okay. So at the end, I close this meeting, this lecture. Okay. Good morning. Have a day for all of you. Okay, bye. Oh, maybe
thank you very much.
Okay, thank you very much, Dr. Paulo and the team, Professor Telvina.
Thank you all.
Professor Sonia Rodriguez also. Thank you.
Bye.
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 Thank you all. Bye-bye.
Bye.
Bye-bye.